This program has been made possible by a grant from the UCF Office of Research and Commercialization.
The UCF Office of Research and Commercialization is committed to moving the discoveries of our faculty and students from ideas to innovation to realization. By moving research from the laboratory to the private sector, we are helping to diversify Florida's economy and helping to bring high-paying jobs to our state. This program presents some examples of our research and our efforts to transition this research to the private sector.
Welcome once again to Zenith. I'm Ed Highland, your guide as we travel across the world of research. In this program, we'll take a look at stem cells, what they are, and why their study will help determine if they have the potential to treat diseases like Alzheimer's. We'll also check on a way that research is being used to help train better teachers. Are our kids getting the science they need? Details coming up on Zenith.
University of Central Florida Technology Incubator, cultivating success through creative ideas, performance, and partnerships.
Because there's a lot of people doing a lot of work every day, and that's what everybody brings together. Certainly, it wouldn't be successful without the partnerships.
The UCF Technology Incubator is a university-driven community partnership providing early-stage technology companies with the enabling tools, training, and infrastructure to create financially stable, high-growth enterprises. With locations in the Central Florida Research Park, adjacent to the UCF campus in East Orlando and in downtown Orlando, the UCF-TI is home to over 50 new and innovative high-tech companies. That's up from 12 companies when the incubator opened its doors in 1999. The incubator has created hundreds of high-paying jobs in Central Florida with an average annual salary of $59,000. Our clients include Nanospective specializes in advanced materials characterization. Utilizing the University of Central Florida Materials Characterization Facility, Nanospective performs nanoscale and macroscopic property evaluation.
We're kind of unique in the sense that we're all UCF graduates. We have firm foundations here at the university with the facility and with the incubator.
The UCF Technology Incubator, promoting optimal corporate growth and making a significant contribution to the economic development of the region's high technology sector.
Many states have testing programs for high schoolers to determine just how much they've learned and if they're ready to go on to the next grade or college. One way to improve the scores is to help teachers teach better with greater comprehension. That's the idea behind the National Science Foundation's Graduate Teaching Fellows in K-12 Education, or GK-12 program.
To a lot of implications on electric charge.
It's a light to illuminate young minds.
Okay, so if I'm here and I double, triple, quadruple, what happens to the amount of shadow that's cast? And then you have to create a mathematical rule that explains it from your data.
Mike McKee's GK-12 program pairs Orange County 9th grade teachers with University of Central Florida graduate students to develop simple ways of making complex science more understandable. The teachers know they start with a handicap in the classroom.
They don't come to class excited. So the toughest thing is getting them excited about what I'm trying to teach. And there are some things, I do teach that a lot of them aren't finding exciting, so I try my best to make it exciting for them. So you can start with the low-tech stuff, or the low simple stuff, and then bring it up to how it is important in their things that they find interesting.
In this instance, low tech is lines on cardboard, a bright light, and another piece of cardboard to cast a shadow. The combination is designed to explain what's called the inverse square law, or to answer the question of how much dimmer a light gets the farther away from it we are.
You'll say the word inverse and they're just like, we don't want to listen to you anymore.
So the challenge becomes translating the science these teachers and researchers work with in the lab.
I mean, they're not going to put it together with the numbers, but as long as they can see that, again, it's inverse.
What'd you guys get?
Into an equation on the high school level.
Like if they recognize a parabola, I think they'll see, as a square relationship.
With the goal of it adding up to a 9th grader who embraces science.
9 graduate students and 1 undergraduate student, along with 10 science teachers, took part in this GK-12 program. Still to come, it's a controversial subject, but could there be secrets to help fight disease locked away in stem cells? A researcher's perspective when Zenith continues. One of the most controversial subjects in politics these days is that of stem cells. Most commonly associated with prenatal cells, stem cells can also come from other parts of the body, such as bone marrow, in what are called adult stem cells. Dr. Kim Sugaya recently gave an overview of his research at the University of Central Florida.
Yeah, all stem cell things already everybody talked about, so I don't have to talk about the stem cells. So my question is, what makes human to be human? How can they behave like a human? So the brain. The tissue transplantation we can do, right? If you have a kidney failure, we can transplant the kidney. So why don't we just transplant the brain? We have a brain bank somewhere, right? But I don't think so. If you transplant the brain, if you have somebody else's brain, that's not you anymore. So I would say brain, the final frontier. We need a brain. But also stem cells, right? I have to talk about the stem cell a little bit. In Florida, so the Alzheimer's disease, that's a tragic disease. Alzheimer's found this one like 100 years ago, and the brain shrunk in the patient brain, okay? And then that makes like this happy couple to become like this. So he tried to reach out to her, but she cannot respond it. And then you might remember, of course, this gentleman. He survived assassination, but he couldn't survive the Alzheimer's disease. And this is the last moment saying goodbye to everybody. I mean, he said, disclosed he had the Alzheimer's disease. How do you feel? So we have to do something. So fortunately, Florida has a fountain of youth in central, and that's where this Ponce de Leon tried to find those in Florida, across the ocean, but in St. Augustine. I went there, but that's not so great, salty tasty water. Okay, so this is the picture I found on the internet calling Fountain of Youth. Right in the middle, you can see like a sphere floating. That reminds me stem cells. So this is a stem cell I isolated from the 9 weeks old human fetus brain. Okay. So we can isolate the stem cell from the human fetus and then create the variety of the brain cells, including the neuron and glia. Here's the neurons, human neurons in vitro, human astrocyte, glia in vitro. Okay. And this is the movie. Actually, after those cells differentiated under the microscope, you can see the large cells, that's astrocyte, and small cells moving along the edge of the astrocyte. That's the neurons. That's what the cells we want to have in our brain. So, although this is in vitro, okay, but this may happen in our brain in vivo. So, we try to do that. We transplant the cells into the rat brain, not the human brain yet. Before that, we did a water maze. This is the water maze. Here you can see the gall, that's a platform, a little bit beneath of the water. So the animal cannot see it. But the animal doesn't want to swim forever. So it's going to memorize and reach there. That's what we call spatial memory. This memory particularly lost in Alzheimer's disease. So we use this one as Alzheimer's model. Okay, so this is the results before transplantation. Young animal, of course, they can reach to the goal pretty good. But some of the young animal, they're okay. They are not so great. Kind of stupid, kind of silly, or whatever. But those changes increase by the age. At the 24 months old, that's the lifespan of this animal. Then you can start to see this group of animals never reach to the goal. Even if we put the animal onto the goal, they kick out from the goal. They don't care, like Alzheimer's. Okay. But some of the animals still okay. They behave like a young animal. Okay, so we mark the cells in vitro and transplant using the stereotaxic and into the brain, right there. And then let it go 4 weeks, and then we do water maze again. So this is before and after. Like this animal, tremendous improvement in cognition. So they can reach the goal very quick, even better than some of the young animal. Surprise. And another interesting thing is all such a stupid young become smart. Some people told me, of course, because we put the human cells into the rat. Yeah, of course. And matter of fact, those animals start to speak Japanese. So this is the statistics, okay? Before transplantation, after transplantation, we saw the significant improvement in cognition. So it works, right? But the control doesn't do anything good. So when we look at the brain of those animals, this green is a neuronal marker, neurons. And this red is the marker for the transplanted cells. So the transplanted cells became beautiful neuron in the cortex and the hippocampus, those areas where we needed such a new cells to make a good memory. And another thing you might notice is the human cells much bigger than the rat cells. This blue are the rat cells, and this green human cells. But still, the beautiful neurons we can make. So maybe those fetal stem cells is the answer to treat Alzheimer's, Parkinson, and so forth. And all the media, they liked it. Bad, always bad. The question would be immune response. So if the cells get the immune response, they chewed up by the immune cells. Another question, anybody wants to have somebody else's brain? I wouldn't. Somebody says if I can get Einstein's brain, that's okay. But I don't know. Like thrill seekers, gambler, or skydiver, they have a little bit different gene. We call it the D2A1 allele. Such a little bit changes. But if you get those cells in your brain, you might start jump from the airplane. You never know. Another thing is do you want to see this happen? You can see in a minute making a fetus from the egg, right? Embryo. And this is a fetus. Because the fetal stem cell is much better than the embryonic stem cell because they know what to become. If we isolate from the brain, they can become brain cells. If we isolate from liver, they can become liver, whatever. So that's another thing, ethical problem. So what we are proposing here is that this is the embryonic stem cell. they can differentiate into the certain type of the stem cells by the environmental cue. Now, for example, I'm taking a neural stem cell and a mesenchymal stem cell from the bone marrow here. And neural stem cell only become brain. Mesenchymal stem cell only become bone, muscle, cartilage, those connecting tissue. So if I put this mesenchymal stem cell in a brain, we're going to have a bone in a brain. All right, that's not good. So my proposal is we just bring them back, de-differentiate, back to the stage, and then bring them to the neural stem cell and make a brain cells. That's one. Another proposal is to just increase the neural stem cell in your brain. You have stem cell in your brain still. Adult, everybody. I hope still I have some left over. Okay. Why don't we just increase those? Those two approaches. One is that this is a bone marrow stem cell. We introduce the gene from the embryonic stem cell. Then we start to see the embryonic stem cell formation here. They can grow a lot. And then we can do the another gene check. All the embryonic stem cell genes start to express. So they look like embryonic stem cell. How about if they behave like embryonic stem cell? So this is the cells. We put such a gene. We put in a basket, which has a membrane in the bottom. And then we plate the human brain tissue, actually the fetal neural stem cell in a culture. And then we put those baskets on the top. That way, those cells almost like being in a human brain condition. So after that, we start to see the cells differentiate into the neurons. This green are the neurons. And the morphology-wise, they looks like a neuron from the embryonic stem cells. This is another picture. And not only that, this one is a little bit different cells, but we treat the cells in a way to de-differentiate and put it in the brain. We can see the neurons in the brain. Hippocampus, we can see the neurons, beautiful neurons. So it means they also behave like a neural stem cell. And not only that, by introducing the certain gene, we can make a dopaminergic neuron. We can make a cholinergic neuron, which can be used for the Parkinson and Alzheimer's disease, and also in the hair cells. If you go to the rock concert, bam, 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 the big noise, see, you're going to damage your inner hair cells. You need this. So that's the thing. Another thing is the stem cell population in the brain. We checked memory impaired animal. I told you, the animal almost like Alzheimer. They don't have any stem cell anymore. This is in the cortex. This is the aged memory unimpaired animal, still memory okay one. They still have two dots, right? You can see that? That's a stem cell there. So maybe the stem cell population in the brain is quite important to maintain a day-to-day normal brain function. But we have a compound which increases the stem cells a lot. Tremendous increase. You can see lots of dots after this treatment. We just inject this compound peripherally, not in the brain. Then we can see such an increase. When we count the number of the cells, tremendous increase, 7-fold increase. So maybe this is good. And when we check the home of the stem cell, that's called subventricular zone, right in the middle of the brain. Okay. And again, aged memory impaired animal, they have much less stem cell compared to the normal unimpaired animal. But after drug treatment, such a compound, tremendous increase, tremendous increase. Not only just the increase, but also they start to migrate out and become neurons. That's good. So I think that's a story. This is my lab, okay? Yeah. Maybe I can stop right here.
Dr. Sugaya has also managed to genetically manipulate adult stem cells so that they changed into specific cell types. Well, we've almost run out of time for changes, but we always save a little room for our Research Minute.
This is clinical microscopy.
microscopy lab. So what we're working out first is they learned how to use the microscopes in much more detail than they would in general microbiology, and now they're looking at human samples of urines and they're examining it for abnormal materials, usually cellular materials, to indicate any kind of renal disorder, bacteria for bacterial infections regarding any part of the renal system, and we can use a urinalysis to pretty much test any metabolic activity of the body. Once they feel comfortable doing urines, our next step would be to get into other body fluids and examine those for abnormal cell types to be able to help identify cancer cells and everything to aid completely in the diagnosis for the patient. So we're helping the physician side by side, even though we're always in the background.
Stem cell discoveries, science for our kids. You never know what you'll find on Zenith. The goal of research is to better understand the world around us. Our goal is to be a window to that world. I'm Ed Highland. Thanks for joining us.